everyone, welcome back. So this is week 9 of the NPTEL course Fundamentals of Protein Chemistry. The course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur, and I'm Beas Mukherjee, a research scholar from Dr. Shomode's lab, the Department of Bioscience and Biotechnology, IIT Kharagpur. And in today's class, we are going to talk about membrane proteins and how they are important in the transport of different materials across the membrane. So in the last class, we had talked about how proteins are important in transport of materials within the cell, and today we are going to talk about membrane proteins in details. Okay, whether it is between different organelles, whether it is between different cells. So we are going to discuss all of that. Okay, let's begin with today's class. Okay. Now let's talk about membrane proteins. So membrane proteins are defined as proteins that are associated or attached to a cellular or organellar membrane. Okay, they are divided into peripheral and integral proteins. Peripheral membrane proteins are temporarily associated with the lipid bilayer, but they do not fully span the membrane. So integral proteins are permanently embedded within the phospholipid bilayer, and they span the entire lipid bilayer and contain hydrophobic alpha helical or beta barrel structures residing inside the membrane. Right, so in the figure you can see that there are different kinds of membrane proteins, and we are going to talk about them in details, okay, in the upcoming slides. So membrane proteins are important signaling mediators in cellular pathways and therefore are involved in diseases like cancer, cardiovascular diseases, and neurological conditions. Approximately 50 to 60% of targeted therapeutics are these membrane proteins, particularly the G protein coupled receptors. Now, when you learn about signaling pathways, you will know that G protein coupled receptors are one of the major group of signaling cascades in our cells, and these are largely targeted by therapeutics, okay, for treatment of different diseases. Now, ion channels, transporters, these are also important drug targets. Okay, now we'll get into this topic, okay. Right. Now, if you recall the fluid mosaic model about which you had learned back in school, then you will remember that there is this phospholipid bilayer, okay, and the lipids are made up of this hydrophobic tail and hydrophilic head. One second. So we'll carry on. Now these lipids, they have a head which is hydrophilic in nature and a tail which is hydrophobic. So the tail, you can see this points inward, and the head remains in contact with the liquid. Okay, that is having different kinds of ions and different kinds of other molecules. Now this can be the cytosol and it can also be the extracellular matrix. Right. So now there are proteins that you can see they are present on these phospholipid bilayer. These are the peripheral proteins, and then there can be proteins that span the entire lipid bilayer, and these are called the membrane proteins, which are integral membrane proteins. Okay, now on to the proteins, there can be small oligosaccharides attached. So you can see them as branching out oligosaccharides, and these proteins have different functions and they have different modifications, and we will talk about them in detail. So these proteins can be glycosylated, so they can be glycoproteins, they can be phosphorylated, and they can have different purposes in the cell. Okay, right. So we have this question for you, in which they are asking which of the following motif is mentioned in the above figure. So what you can see is there are different beta sheets, and the beta sheets are arranged in such a way that it gives the structure of a helix, right? So then we look at the answer, and the answer is that this is actually a beta helix, which is formed by association of parallel beta strands in 2 or 3 faced helical pattern, resulting in the tandem protein repeat structure known as beta helix. And this particular protein, which is called acyltransferase LPXA, found in E. coli, this has this kind of a structure. It's a left-handed beta helix. Okay, now we have another question. Okay, so in this question, it's a simple and very straightforward question where they are asking you to point out which is X and which is Y, which is a peripheral membrane protein and which is an integral membrane protein. Now, this is a very straightforward question. So, we look at the answer based on the fluid mosaic model. We'll be able to answer this. Right. So, we come to the answer. We see that X is your integral membrane protein and Y is the peripheral membrane protein. So integral proteins are membrane proteins that are embedded within the membrane permanently. Peripheral proteins are the membrane proteins that adhere with the membrane temporarily and interact with the membrane indirectly by interaction with the integral protein or directly by interaction with the polar head group of lipid. Okay. Now, we'll talk about some of these membrane proteins and their functions in general. Okay, so now membrane proteins can be of different types. For example, there can be these ABC transporters. There can be ion channels. Okay, and there can be membrane bound ATPases. ATPases are helping in ATP generation. And then there can be SLC transporters, and there can be water channels. So water channels help in transport of water molecules. Okay, so now they can have different kinds of activities. For example, enzymatic activity. Now the procession of metabolites and substrates for various metabolic pathways. They can help in signal transduction. So chemical messengers interact with the membrane protein binding sites to send forth signals. Okay, then there can be transport of materials. Now this can include active transport and passive transport. Now you know that the difference between active and passive transport is that active transport actively uses ATP in the form of energy for this transport. Okay, and it is often not in the direction of higher concentration to lower concentration. It can also be in opposite direction. That is why ATP is being used. And for passive transport, this is always down the gradient. That means higher concentration to lower concentration transport is happening, and this is not using any energy as such. Okay, so that is passive transport. Now molecules and other substances move across the cell membrane using these mechanisms. Then we come to cell-cell recognition. So identification between cells is very important aspect of the immune system, right? So that is done by looking at the different cell surface markers. So these different cell surface proteins are important characteristic of a particular cell, and they symbolize whether a cell is, let's say, cancerous, whether a cell has this particular receptor protein, and then you will know that it is a B cell or it is a T cell, Okay, so there are different immune cells. That is how you get to know whether this particular cell is a particular kind of cell. Okay. Now there are intercellular joinings, which means that different junctions such as gap junctions and tight junctions connect the neighboring cells. The sixth one is anchorage or attachment. So important for cytoskeletal network, protein locations and uphold of certain shapes. So these anchorages are important for holding the shape of the cell and also cytoskeletal network formation. Okay, now we will look at all of these in a little more details. So for example, ABC transporters. Now what are ABC transporters? These are ATP binding cascade transporters. That is where the name ABC comes from. Okay, ATP binding cascade transporters. Now these are proteins that use ATP hydrolysis to actively transport substrates such as sugars, ions, lipids, peptides, drugs, etc. across the membrane, often against the concentration gradient, the one that I was telling you right now. Okay, so this is transporting all of these different materials against the concentration gradient using ATP hydrolysis. Okay, now the next one is ion channels. Now, ion channels help in the transport of different ions such as sodium ion, potassium ion, calcium ion, chloride ions, etc. So, they are participating in passive diffusion of ions across the membrane down their electrochemical gradient or concentration gradient, crucial for nerve impulses, muscle contraction and signaling. Okay. Now, the next one is membrane bound ATPases. Now, these can be of different types. There can be P type, there can be F type. There are also other types like V type, and you can read about these in details. Okay. Excuse me. So, coming to the F type. F type ATPases, Na plus K plus ATPases. So, couple ATP hydrolysis or synthesis to ion transport, and F type ATPases are important for synthesizing ATP in mitochondria and chloroplast. So, if you remember the electron transport chain that was there in the mitochondria, you would remember that there was this F0, F1 particle, right, in the ATP synthase, which is also membrane protein. So, that is an F type. So, F type is having this F0 and F1 structure. Okay. Now, see, Na plus K plus ATPases, they maintain ion gradient essential for membrane potential. So, these are the different types. You need to remember which is what, and there can be questions from here. Okay. Now, we move on to SLC transporters or symporters. Now, what is symporter and what is antiporter? Now, this is very interesting. So, symporter is a protein which transports another substance with our substance of interest together in the same direction. Let's say this is the protein, something binds here and another thing binds here, and this just turns. So both the things get transported to the same direction. If it's an antiporter, then what will happen? This is the protein, something binds here, and then when it flips, these two are getting transported to two different directions. Right, so that is an antiport. So see, what is a symporter? Example of a symporter is sodium glucose symporter. So what happens is, when glucose is supposed to enter into the cell, sodium is often entered along with the glucose molecule. Okay. So moves two different molecules in the same direction across the membrane. Example, glucose uptake with sodium. Then there is antiporter. So example is sodium calcium exchanger. Here what happens is, this moves two different molecules in opposite direction, that is exchange of intracellular calcium for extracellular sodium. Okay. Then there is something called uniporter. Uniporter is transporting only one kind of molecule at a time, one molecule. So for example, glued transporters, these transport a single type of molecule down its concentration gradient using facilitated diffusion. Now we come to water channels. that is aquaporins. Now these are specialized protein which only help in transport of water molecules, rapid and selective transport of water molecules across the membrane, maintaining osmotic balance. So these are the different things that are going on inside the cell membrane, and these are all helping in transport of different molecules across the membrane. Okay. Right. So now we have the process by which proteins are targeted to a specific organelle is called protein sorting. Okay, now we are going to talk about something very interesting. And what happens in protein sorting is that it can be of two types: signal-based sorting and vesicle-based trafficking. Okay, so membrane proteins are sorted into vesicles by the COPII machinery and delivered to Golgi complex by vesicle tethering and SNARE machineries. Now what are these? So if you look at the figure, you will see that proteins are synthesized either on the free ribosomes or on the ribosome that is bound to the endoplasmic reticulum. Okay, so now what happens is the polypeptide synthesized with the chloroplast transit sequence. So that means what happens is once the protein is synthesized, the protein can undergo two different things. It has a signal sequence, right? So let's say that this signal sequence is for chloroplast. So there is a sequence of residues which says that this protein is supposed to go to chloroplast. Now what happens is the polypeptide synthesized with the chloroplast transit sequence and secondary targeting sequence. So what is the secondary targeting sequence? You will see that these proteins are sometimes carried off to a particular place, and the polypeptide synthesized with mitochondrial pre-sequence. So there can be different kinds of signal sequences. Okay, polypeptide synthesized with C-terminal PTS signal which is going to the peroxisome. Then there can be transit sequences that are cleaved before the protein is folded in the stroma, or secondary targeting signal directs the protein to the final location. So now the protein is going to your target organelle, and there the signal sequence is getting cleaved, and that is how these proteins are actually transported. Now we look at this in details. Okay, now this is actually called the signal based sorting. That means as the protein is getting produced, the protein is already having the signal sequence, and because of the signal sequence it is directly getting transported to the respective organelle where it is supposed to go. Now another thing is for the proteins that are produced on the ribosome attached to the ER membrane, we look at that. Okay, so first we will talk about this a little bit. So see, there can be post-translational transport and there can be co-translational transport. So what are these two things? So what is post-translational transport? This means that the protein is completely synthesized. So from the mRNA, the protein is completely synthesized. It is having the signal sequence, and now it is getting transported to different different organelles. For example, mitochondria, peroxisome, chloroplast, nucleus, etc. Right, so this is called post-translational transport. That is, they are getting transported after the completion of the translation process. Then there can be something called the co-translational transport. That means as the protein is getting synthesized, it is being sent somewhere using vesicles, vesicular transport mechanism, right? So for the proteins that are synthesized in the ribosome that is attached to the rough endoplasmic reticulum, what happens is the proteins, as the proteins get formed, they are either entering into the ER if they are having this sequence for ER, they are either entering into the ER or they are getting packaged into vesicles and they are traveling to different places. For example, the Golgi apparatus. So from ER, the protein is going into Golgi, and from Golgi, it is going to different places like the cell surface. If it is an exterior cell exterior, if it's a secretory protein, it can go to the plasma membrane, it can go to the lysosome, it can go to other endomembrane system. Okay. Now for import into different organelles, what should be the signal sequence? Okay. So So for input into nucleus, the signal sequence is proline, proline, then 3 lysine and then arginine, lysine, valine. So basically from here what you have to do is you just have to remember which residue is getting repeated. Okay, so that is something that you have to remember. For example, for going to the, let's say, import into nucleus and export from the nucleus, what is the majorly repeating residue? So these can come as questions for exams. Okay, that is how there are signal sequences for different organelles, and these are given here for your reference. Next, we are going to talk about this post-translational and co-translational transport in a little more details. So see what happens in co-translational transport. So ribosomes are attached to the ER membrane if they are synthesizing polypeptides destined for the endomembrane system. So what is endomembrane system? Endoplasmic reticulum, Golgi, and then lysosome, and then the other different things, for example secretory vesicles and plasma membrane. All of these make up the endomembrane system, and this the proteins that are going to the endomembrane system, they are all synthesized by co-translational co-translational trafficking. Okay, so ribosomes attach to the ER membrane if they are synthesizing polypeptides destined for the endomembrane system or for export from the cell. Now, as synthesis continues, a newly forming polypeptide is transferred across the ER membrane by co-translational import. And what happens is the completed polypeptide either remains in the ER or is transported via vesicles to another compartment of the endomembrane system, that is integral membrane proteins are inserted into the ER as they are made rather than being released into the ER lumen. Then the signal sequence, whatever is there, that is getting chopped off and then it is getting transported to the Golgi. From the Golgi, it is undergoing proper folding, and then there are a lot of different checkpoints to ensure that the protein is folded properly, and then it is sent off in the form of, they are wrapped around by vesicle and they are sent off to different places. Okay. Now, what happens if it is post-translational transport? So, for post-translational transport, see, ribosomes remain free in the cytosol if they are synthesizing polypeptides destined for the cytosol or for import into the nucleus, mitochondria, chloroplast, or peroxisomes. So, when the polypeptide is complete, it is released from the ribosome and either remains in the cytosol or is transported into the appropriate organelle by post-translational input. Polypeptide uptake by the nucleus occurs via nuclear pores using a mechanism different from that in the post-translational uptake by other organelles. So what is happening is the completed polypeptide is there in the cytosol and it either remains in the cytosol. If it is a cytosolic protein, it can go via nuclear pores into the nucleus. It can go to the mitochondria, to the chloroplast, to the peroxisome, or it is inputted to an organelle, okay, depending upon the signal sequence. Okay, now we'll talk about this transport across the endomembrane system in a little more details. Okay, so if you see the figure, you will see that this is the place where the rough endoplasmic reticulum is there, because the ribosomes are attached to the wall of the rough endoplasmic reticulum. Now what happens is the protein is exiting in the form of a vesicle, and this vesicle is going to the Golgi. Okay. Now the Golgi has different layers, so we'll talk about that. So in the secretory pathway, that is black arrows, cargo proteins start their journey by budding off the ER exit sites, that is the ribosome free areas of the endoplasmic reticulum. So these proteins are packaged into the COP2 coated vesicles, which later fuse with the endoplasmic with the vesicular cluster, that is endoplasmic reticulum Golgi cluster. Several ER GICs merge and fuse to form the cis Golgi network. So here this is the cis Golgi network, and next what happens is COP1 vesicles mediate the recycling of ER proteins and are also thought to mediate the transport of cargo between the Golgi stacks. So once in the trans Golgi network, so initially the Golgi that faces the ER, this is the cis Golgi network, and then between the Golgi track it is going to travel, and then it is going to the trans Golgi network, which is facing the cell membrane, okay, or the plasma membrane. So that is your trans Golgi network. Now see what happens is, once in the trans Golgi network, the proteins get sorted to the plasma membrane, or in some cases to lysosome, if they are, let's say, a protein important in the lysosome or any other purpose. Okay, so resident proteins of each organelle along the secretory pathway achieve their localization through specific retrieval mechanisms, which is the red arrows that are shown in the figure. So see, different types of coated vesicles and tubulovesicular carriers transport cargo from the trans-Golgi network to the final destination. For example, some cargoes reach the plasma membrane and or cell exterior by means of secretory granules, while others appear to travel in tubulovesicular structure or through recycling endosomes. In the endocytic pathway, which is shown by blue arrows, molecules are internalized in the form of plasma membrane derived clathrin coated vesicle. Now this is very important because there will be questions from here. Okay, so clathrin coated vesicles, you can see the blue arrows there. Now these vesicles are derived from early endosomes and then to late endosomes and eventually to the lysosomes, where their content is typically degraded. Okay, and let's say that some protein is not folded properly. Now there are a lot of different checkpoints in this entire pathway. So if even after repeated trial the protein is misfolded, then it is sent for these degradation pathways. Okay, then what happens is their content is typically degraded. So retrieval pathways shown by red arrows allow for the recycling of the protein from the early endosome to the plasma membrane surface and from the late endosome to the Golgi. So these can once again come back to the ER once again, go through the Golgi, and that is how the entire process happens. Okay. Now we'll talk about these vesicles in a little more details. Now I am going through this in such details because this is one of the favorite topics of your examiners. So there can be a lot of questions from here, and that is why it's very important that you remember what are COP1 vesicles, what are COP2 vesicle, what is cis Golgi network, trans Golgi network, how this entire vesicular transport of membrane proteins happen. Okay, so see, a generic post-translational pathway for protein trafficking to the plasma membrane. So after synthesis in the endoplasmic reticulum, membrane proteins are sorted into vesicles by the cotamer protein complex 2, which is your COP2. Okay, machinery delivered to the Golgi by vesicles, tethering and SNARE machineries. Now, intra-Golgi transport and retrograde transport from Golgi to ER. So, this is anterograde transport from ER to Golgi. This is mediated by COP2 vesicles using tether and SNARE. And the retrograde transport and the transport between the Golgi membrane, Golgi network, that is mediated by COP1 vesicles. Okay. Now, they are sorted into vesicles by intrinsic sorting motifs. And okay, so that is done. Okay, now at the trans Golgi network, so that means the protein is definitely folded properly and it is going to be sent away now to the different membranes. So at the trans Golgi network, the proteins are sorted into vesicles by intrinsic sorting motifs and cytoplasmic adapter complexes and are transported along the cytoskeletal elements to the plasma membrane. So protein delivery to the plasma membrane is mediated by vesicle tethering and SNARE machineries. Some proteins, for example, ligand receptor complexes, are internalized through another set of adapters and delivered to the endosome, from which they might be recycled back to the plasma membrane. Okay. So it's very important that you read about this in details, and I would recommend the book Bruce Alberts, from where you can read. Okay, so all the links are given at the end of the PPT for your reference. Now this is the kind of question that you can expect. So see, here is a sequence given to you, amino acid sequence. So this is present in the dash of a globular protein. So you have to tell me whether this is present in the interior hydrophobic core or it is present in the exterior hydrophilic surface. So if you look at the residues, you see that most of these are made up of hydrophobic residues, right? So now hydrophobic residues are more likely to be in the hydrophobic core of the protein for proper folding and stability of the protein. And since this is a globular protein, it is definitely soluble in water. So that means the surface of the protein will have more hydrophilic residues and the interior of the protein will be packing the hydrophobic residues very well. That is why, what should your answer be? The answer is B, interior hydrophobic core. So hydrophobic effect is the major mechanism by which globular proteins preserve their tertiary structure. Non-polar residues are clustered in the hydrophobic core and the charged and polar residues are present in the hydrophilic surfaces. So these amino acids mentioned here. So these amino acids are generally non-polar, and that is why they are present in the interior hydrophobic core. So hello, so in this class we are talking about membrane proteins, okay, and let's try to solve a few questions. Okay, now we are talking about transport proteins that are important, membrane proteins, okay. So the first one is membrane channels. Membrane channels allow free transport of ions or small molecules along the concentration gradient. So along the concentration gradient means that there is passive transport by the method of passive diffusion, and channels specific for passage of water are lined by aquaporin molecules. Aquaporins are definitely important for the passage of water. So membrane pumps. Membrane pumps are involved in active transport of molecules against their concentration gradient. So these are the different ATP, Na plus, K plus, ATPase pump and membrane carriers. So membrane channels, membrane pumps and membrane carriers. These are the three types of things that we came across. Membrane channels include transport of ions, okay, and aquaporins. The membrane pumps are using ATP for transport of molecules up the concentration gradient, against the concentration gradient. And then there are membrane carriers, which are uniporters, symporters and antiporters. Okay. Now we have already discussed about these uniporters, symporter and antiporter, right, previously. So here we are going to talk about diffusion channel proteins and the gated channel proteins and membrane pump. So see what happens is diffusion. So water molecules definitely they can diffuse in using aquaporins. Then there are ions. For ions there can be passive diffusion. Then the channel proteins again useful for so water molecules there can be just normal diffusion. Then there can be transport across the aquaporin, which is a channel protein, membrane channel protein. Okay, similarly ions can also be transported using these channel proteins. Next there is gated channel proteins, and there are the membrane pumps which use ATP for the transport of ions against their concentration gradient. Okay, we'll look at these in a little more details now. So what are these gated channels? Now gated channels can be of different types. They can be ligand gated channels, they can be mechanically gated channel, they can be voltage gated channel, and there can be these channels which are always open. So leaky channel. Okay. So now what are these? So ion channels are integral membrane proteins that only allow ions of a certain size and or charge to pass through the cell membrane through passive transport down the concentration gradient. Okay, so passive transport is only getting allowed. So they are essential for various physiological processes using signaling, muscle contraction and maintaining cellular homeostasis. Okay, so ligand gated, that means let's say a particular ligand is coming and binding to it. Only when that ligand is binding, let's say it's a neurotransmitter. So only when that is binding, the channel is opening and it is allowing sodium to pass through. Okay, it's like that. The next one is mechanically gated channel. So that means only when there is a specific amount of pressure in the cell, it gets closed or it gets opened, and because of which the ion can either pass through or not pass through depending upon the pressure. Then there is voltage gate. So let's say some electrochemical voltage is generated across the membrane. Because of that, it can either open or it can close at the passage of the ion is allowed or not allowed. Some are leaky channels. They are always open and they are allowing only certain kinds of ions to pass through. Okay. Right. So now we will go to the next slide. So see, everything is given in details for you. Okay. So voltage gated channels open or close in response to changes in the membrane potential. They are involved in the action potential and nerve signaling. Then there can be ligand gated channels. They open in response to the binding of the specific ligand. For example, neurotransmitter. So these help in mediating synaptic transmission and muscle contraction. Then mechanically gated channels. So they open in response to mechanical stress or deformation of the membrane. Play roles in touch sensation, hearing and proprioception. Then there is leaky channels. So what are these leaky channels? They are always open, allowing for passive flow of ions. And they help in maintaining the resting membrane potential of the cell. Okay. Then we come to calcium channels. Calcium channels are specialized for calcium ions and can be voltage gated or ligand gated channels. They are very important for muscle contraction, neurotransmitter release and various signaling pathways. Okay. Now we'll talk about targeting these different membrane proteins for our purposes, our different purposes. Right. So these are done using different kinds of drugs. Now, a group of these drugs can be antibiotics. Now, what are antibiotics? Antibiotic means that a particular living organism is producing some kind of a substance which is either killing or inhibiting the growth of another group of living organism. So that is antibiotic. Okay. Now, antibiotics can work at different levels. So, they can work at the DNA level, protein level, RNA level, at different levels. Okay. So, now there are certain antibiotics which target membrane proteins. And you saw that around 50 to 60% of the drugs that we have today, they are all targeting these membrane proteins. Okay, GPCRs. So, we will look at them in details. So, see. Antibiotics, the term antibiotics means against life. Antibiotics are organic compounds produced by one organism that is toxic to another microorganism even at low concentration. However, modern antibiotics also are made partly or wholly through synthetic means. So, antibiotics inhibit or kill microorganisms, more specifically bacteria, through various mechanisms acting on different cell sites. The mechanism of action of antibiotics varies broadly. Okay. So now antibiotics can impact cell wall synthesis. So they are the beta-lactam antibiotics. Then there can be nucleic acid synthesis can be interrupted. For example, DNA guidase activity can be interrupted, and RNA polymerase activity can be interrupted, and then protein synthesis can be hampered. So 50S ribosome subunit can be targeted, 30S ribosome subunit can be targeted, and the cell membrane can also be targeted. Okay, so these are the different ways in which antibiotics attack harmful bacteria in our body. Okay, so we look at all of these different types, and you have to remember the names of these antibiotics and their mode of action, because these can come in the form of match the following. These are very important for your exam. Okay, so some of the different classes of antibiotics are given here. So see, the antibiotics targeting bacterial cell membrane, they are normally polymyxin. So what is this polymyxin? Polymyxin binds to the lipopolysaccharide and phospholipids in the outer membrane of the gram-negative bacteria. So lipopolysaccharide is there in the cell wall of the bacteria and phospholipid is there in the cell membrane of the bacteria and disrupts the membrane integrity, causing cell lysis. Then there is lipopeptides. So lipopeptide binds to the cell membrane in a calcium dependent manner, disrupting membrane potential and inhibiting protein, DNA and RNA synthesis. Then there can be bacitracin, which is very commonly used antibiotic. You might have heard about this. So bacitracin inhibits dephosphorylation of the lipid carrier involved in the cell wall synthesis, affecting cell wall integrity and leading to lysis. The next one is streptomycin. Streptomycin primarily inhibits protein synthesis but at high concentration and can disrupt membrane integrity, increasing permeability and causing cell death. Okay. The next one is tetracycline. So tetracycline inhibits protein synthesis with membrane disruption at high concentration, leading to cell death. So tetracycline, streptomycin, bacitracin is a very important and commonly used antibiotics. It's good if you remember these. Okay, now we have the mechanism of action of antibiotics targeting the cell membrane. So see what is happening here. Mechanism of action of gramicidin A. Gramicidin monomers form beta helix conformation within the membranes. Dynamic dimerization of two monomers form the functional channel, which consequently induces local membrane deformation. The cells maintain a low concentration of intracellular Na+ and high concentration of intracellular K+ relative to the extracellular environment. Formation of the gramicidin channel, that is green cylinder shown in figure A, disrupts this balance by permitting passive diffusion of these cations along their respective concentration gradients. It is shown using arrows, resulting in Na+ influx into the cell and K+ efflux. So K+ is leaving, resulting in membrane depolarization, osmotic swelling and cell lysis. Okay, and the structure of gramicidin A is given for you, and this is the kind of beta helix that it forms. So that is the mechanism of action of gramicidin A. Now that you have read about this, you will have to answer this question. Okay, choose the incorrect statement about gramicidin A. It works as an ion channel type ionophore. It is a 15 residue linear peptide. It is a ribosomal protein. It consists of alternating D hydrophobic and L hydrophilic amino acids. And the correct option is which of the following? So we'll have to look at this. Now gramicidin A is a non-ribosomal 15 residue long ion channel protein which consists of alternating D and L hydrophobic amino acids. So if we go back to the figure, we'll be able to see that. Okay. Okay. Now ATPases as membrane proteins. We have a few more slides to cover. Okay. So ATPases are very important membrane proteins. You already know their function. We'll look at the different types of ATPases. Okay. So ATPase pumps use energy in the form of ATP to transport ions against their concentration gradient by active transport. Now essential for maintaining Na+, K+, Ca2+ concentration gradients. Okay. Now there are different types of ATPases. What are these? There is P-type ATPases. Now P-type ATPases are single polypeptide with multiple transmembrane domains, and they are important for transport of ions across the membranes. Then there can be V-type ATPases. So why is it P-type? Because phosphorylation site is there for on the aspartate ion. Okay, so there is a phosphorylation site, that is why. Next, there is V-type ATPases. So V-type is made up of V0 and V1 domain. So V0 domain is actually membrane bound it's a membrane bound domain, and the V1 type is actually a catalytic domain. So V0 forms the proton channel and V1 catalyzes ATP hydrolysis. So proton pumps that are there in the organelles are important V-type ATPases. And then there is the F-type ATPase. So similar to V-type, it has F0 and F1 particle, where F0 is the membrane spanning region and F1 is the catalytic domain. So F0 allows proton flow and F1 synthesizes ATP from ADP and inorganic phosphates. This is the difference between V-type and F-type. V-type catalyzes, right, ATP, but F-type synthesizes ATP. So synthesizes ATP using the proton gradients. The next one is ABC transporters. Now what are these? They are composed of membrane domains and nucleotide binding domains. ABC transporters. And transmembrane domains transport the substrates and nucleotide binding domains remains for hydrolysis of ATP. Okay, transport a wide range of substrates, for example, ions and drugs. Okay, now the mechanism of action is given here for your reference. Okay, so ATP synthase, how does it work? F0, F1 ATP synthase uses a transmembrane proton gradient to drive the synthesis of cellular ATP. So see how is it working? So mechanism of action, the proton, this operates through chemiosmosis. Mechanism of action is that the proton gradient is established by electron transport during the cellular respiration and photosynthesis. Proton flow is happening. Protons flow back through the F0 domain, driving rotation. Rotational catalysis happens. So here what happens is this rotation induces conformational change in the F1 particle. So rotation of the F0 results in conformational change in the F1 particle, facilitating ATP formation. And in F-type ATP synthase, which is found in bacteria, mitochondria, and chloroplast, and V-type ATP synthase is mainly primarily involved in proton transport, okay, across the organelles. Now we look at a question. All aerobic organisms require dash to produce the basic energy by generating ATP, which is adenosine triphosphate, by ATP synthesizing enzyme called dash through a process called dash transport. So we look at the answer. The answer is A, oxidative phosphorylation, ATP synthase and electron. So see, all aerobic organisms require oxidative phosphorylation to produce their basic energy by generating ATP by an ATP synthesizing enzyme called the ATP synthase through the process of electron transport. Now we'll talk about photosynthesizers for therapy. Okay, so a photosensitizer is defined as a chemical entity which, upon absorption of light, induces a chemical and physical alteration of another chemical entity. So a good photosensitizer should absorb photon efficiently, high absorption coefficient, and have high quantum yield of triplet formation, and the triplet state should be long lived in order to have time to react with the neighboring target molecules. So most compounds that form triplet states that are able to produce radicals and reactive oxygen species have a tricyclic, heterocyclic or porphyrin ring-like structure, okay, with conjugated double bonds. That's a pi-electron system. Now we had talked about heme in our previous class, which also has this porphyrin ring-like structure. So what happens is, when heme is subjected to light and a substrate, then no photochemical reactions happen and heat is released. But heme, now heme is made up of iron and porphyrin, And only the porphyrin ring, when it is subjected to light and substrate, what happens is reactive oxygen species are generated. So porphyrin ring consists of 4 pyrrole subunits linked together by 4 methane bridges. Tetrapyroles are naturally occurring pigment like heme, the prosthetic group of hemoglobin, cytochromes, catalase and peroxidase, vitamin B12 and chlorophyll. These tetrapyroles do not induce any photochemical or photobiophysical reactions in other compounds, or are rapidly quenched in their normal surroundings, example chlorophyll. So by removing the metal, tetrapyrrole become efficient photosensitizers. The coordinated metal ions increase the probability of non-radiative decay of the triplet state. Porphyrin-based photosensitizers are used for photodynamic therapy for cancer treatment and age-related degenerative diseases. Okay. So see, this is shown to you in the form of a figure. So typical UV visible absorption spectra has a SORET band and Q bands. Now what are these? Now the SORET band comes at 400 nanometer wavelength and the Q bands are there around 600 nanometer or so. Okay, so the SORET band or the B band at 400 nanometer, that is because transfer from S0 to S2 state, and the weaker Q band at 550 nanometer is S0 to S1 transition. Now S1 and S2 are the second excited states of the molecule. Fast internal conversion between S2 and S1 happens, it's very fast, and B and Q bands both arise from the pi to pi star transitions and can be explained by considering the 4 frontier orbitals of the porphyrin. So now we have a question. Now why we discuss this? Of course that is because you have a question based on that. Okay, so what is a SORET band? So SORET band is mostly due to dash transition, and it is useful to characterize the dash prosthetic group. So of course you know the answer is pi to pi star and heme. So SORET band occurs when due to electron dipole movement which causes pi to pi star transition in the porphyrin compound. Heme is a porphyrin compound. Okay. Now which of the following types of ATPases undergo reversible phosphorylation to transport cations across the membrane? A, F, B and P type. The second question is, bacterial purines are beta barrels. Which amino acid would you expect to be particularly abundant at the entrance of a beta barrel that is specific to phosphate ions? Now this is something that you'll have to think about. Okay, so we have the answers here. So see, P-type ATPases are reversibly phosphorylated. Phosphorylation P-type, okay? Reversibly phosphorylated by internal phosphorylation during the transport of cation. Transport cycle of cations, okay? Now the next one, as phosphate ion is negatively charged ion, to transport an anion through the beta barrel, positively charged amino acids are required. And which of these are positively charged amino acid? Only option A has both positively charged amino acid. Should be present at the entrance of the beta barrel, and hence lysine and arginine amino acids are present abundantly in the entrance of the beta barrel. Now there's another question. Okay, so we have one more question. Indicate whether the following compounds are likely to cross a membrane by channel or transporter. Calcium ion, water, glycine and glucose. So, which of these are going to cross by a channel or transporter? Okay, based on the order of likelihood. So, the answer is A. Now, an ion channel is a pore-forming membrane channel that allows ions to pass through the channel according to the concentration or electrochemical gradient. So, transporters refers to the transmembrane protein that transport ions, small molecules across the cell membrane against the concentration gradient by active transport. Okay, so 1 and 2 are supposed to transport using channel proteins, and 3 and 4 are normally going to get transported by the membrane transporters. Okay. If the substance is uncharged, what information would you need to predict whether the transport will be thermodynamically favorable or not? So information about the pH gradient, information about the membrane potential, information about the solubility of the substance in a non-polar core of the membrane, and information about the concentration gradient. Okay. So which one do you think would be the answer? So of course the answer is information about the concentration gradient, because if delta G is negative, then it will be thermodynamically favorable. And delta G is RT ln ion in by ion out plus zeta potential, and zeta is the charge of ion as the substrate, and then there is the Faraday and change in membrane potential. Okay, so that is the entire equation. So as the substance is neutral in nature, the second term to this equation will be 0. So the zeta potential and this entire term becomes 0. Therefore, information on the concentration gradient is required for the calculation of Gibbs free energy. Right. So now we go to something called the Nernst equation. Now this is very important because there will be a lot of questions based on this. So the Nernst equation is often used to calculate the cell potential of an electrochemical cell at a given temperature, pressure and reactant concentration. The Nernst equation can be used to determine the cell potential of a cell under non-standard conditions. So the equation is given here. E is equal to E naught minus RT by NF to ln Q. So where Q is the reaction quotient, R is of course the universal gas constant, N is the number of amount in moles of electron transferred, and F is the Faraday's constant, and T is the temperature in Kelvin. So E is equal to RT divided by ZF ln CO by CI, where concentration out by concentration of the ions inside the cell, and Z here is the charge of the ion. Okay? So that is the entire equation. So the Nernst equation is useful in calculating the ion equilibrium, potentials, informing the membrane potential, action potential, muscle contraction, ion transport mechanisms, etc. Now there's a question on this. So they are asking you to calculate the Gibbs free energy for the oxidation of 2 moles of NADH by oxygen, and Faraday's constant is given to you. So what is the answer? Let's see. The relation between Gibbs free energy and the standard electrochemical potential is delta G naught is equal to minus NF delta E naught. In the equation, N is the number of electrons involved in the reaction and F is the Faraday's constant. So N is 2 and delta E naught is equal to 2 into your universal gas constant minus, sorry, not universal gas constant, this equation is given here, and then you get Faraday's constant into delta E naught, and so the answer comes as minus 436 kilojoules per mole. Now the next question is on proteins. So this is something that you have read in school. What is a hypotonic, what is an isotonic, and what is a hypertonic solution. So what happens in a hypertonic solution? Hypertonic means that the salt concentration varies in such a way that what happens? Let's say that a cell is placed in an isotonic solution. Then what is going to happen? The amount of water entering the cell is going to be equal to the amount of water leaving the cell, because the solute concentration, ion concentration is going to be equal inside and outside the cell. So the cell is neither going to shrink nor going to swell. But if you keep the cell in a hypotonic solution where the water content is more outside compared to that within the cell, so the ion concentration is more inside the cell, then what will happen is more of water will enter into the cell, and that is going to happen through diffusion. And in a hypertonic solution, the cell is going to shrink because the water in the cell is going to leave, because the ion concentration is more outside the cell compared to that inside the cell. So if you look at the answer here, in hypotonic solution is the one that has lower solute concentration than the solvent concentration. On the other hand, in the hypertonic, it has a high solute concentration relative to the solvent, and isotonic is defined as a solution that has the same concentration of solute and the solvent. Okay. Next, we go to the next slide. So, which of the following are indicative of passive transport? So, ATP is required for transport, and small organic molecules will diffuse based on the molecular weight and solubility. In lipids, molecules are transported from low concentration to high concentration. They are transported from high concentration to low concentration. So, which of these are indicative of passive transport? So, two of these are correct and two of these are wrong. So, if you look at this carefully, you will see that, of course, 2 and 4 are correct, but 1 and 3 are wrong. Why? Because in passive transport, firstly, no ATP is required. Second, the molecules are transported across their concentration gradient and not against it. So, in passive transport, all molecules flow through the membrane from higher concentration to lower concentration according to the molecular weight and solubility. In the lipids, according to the concentration gradient. Okay. There's one more question for you to answer. At 37 degrees centigrade, the internal cell concentration of sodium is 20 millimolar and the external concentration is 340 millimolar. Calculate the minimum membrane potential required to transport sodium out of the cell. So, using this Faraday's equation, we will be able to solve it, and the delta E is equal to minus 75.7 millivolts. That is the membrane potential that is required. And the formula that you're supposed to use is mentioned here. Okay. So that's all for today's class. These are the different books and the different web sources that you can refer to. Okay. And since there are no students today, I'll end the session here, and the recording will be available on YouTube, and also the PPT and everything will be provided to you in the same folder as always. So, thank you for today's class.